welcome to this KeyGene webinar about data science for Genome Insights. My name is Roeland van Ham. I'm head of KeyGene's Bioinformatics department, and today I will be your host for this seminar. We are very happy to have more than 400 registrations from all over the world. And we are very proud to have two great guest speakers for you today, Andrew Carroll from Google Health and Camille de Jong from Ontura. A warm welcome to both Andrew and Camille. During this one-hour webinar, you will get 4 short presentations that highlight our progress in genome data science and the perspectives ahead. At the end of the program, we will have a Q&A session for all contributions, so plenty of time to answer questions that you may have, and we hope to get many. We ask you to kindly type your questions in the chat box, and we will address them after the presentations. So it's my pleasure to give the first presentation in which I will highlight some of the work that we do at KeyGene in genome data science. Before diving into it, let me start by telling you a little bit about the vision and the drive that we have to work in this field of data science, not only for genome insights, but in general for crop innovation. We all know the very significant challenges that our world faces today. Population growth, climate change, pressure on land and water resources, and unfortunately, unequal distribution of food and resources over regions of the world. And that brings a challenge. That the demand for food continues to grow very rapidly, and that at the same time we need to match that demand with a greatly increased supply of food that we must produce in a very sustainable manner, meaning that we need to use far less or even no use of pesticides, fertilizer, and natural resources. And this is often phrased as we need to produce more with less. And despite the fact that we have made great progress in plant breeding and food production over the past decades, we believe that this gap that is opening up in front of us is extremely real, and that at the same time we are starting to experience a little bit this law of diminishing returns in crop improvement. For example, it is estimated that we need a doubling of the world food production in the next 30 years to avoid this crossing point in this graph in the near future. And knowing that the low-hanging fruit in crop improvement has been harvested, we increasingly are facing this highly non-linear complexity in the genetics of the traits and the crops that we want to improve. And it is here that we at KeyGene strongly believe that data-driven innovation in breeding and crop improvement is a crucial and absolutely necessary part to this technological and socio-economical puzzle that we need to solve to secure a sustainable food production for the generations to come. And it is here that we live our passion about helping other agro-food companies and organizations in the world to bridge that gap. We do this by organizing our research and innovation in what we call innovation platforms. 3 of these provide more technological foundations with our digital phenotyping platform, our cell and tissue design technologies, and our Genome Insights platform. 3 others are oriented more towards the crop innovation and development itself, and they make use of these foundational technologies. So today we focus on Genome Insight. But it is good to realize that this data science is becoming impactful for all kinds of crop innovations endeavors. As this is also the first presentation of this afternoon, let me use this to, for those in the audience who do not really know what data science is, to explain this a little bit further. It is often seen as a discipline at the crossroads of, first of all, math, and statistics, and domain knowledge. And this is, of course, where much of the traditional science has produced its output in the past centuries, in fact. At the other side, we have at the crossroads of domain knowledge and computer science. That's where many of the software, domain-specific software, has been produced. And among the upper two, you have this very exciting field of machine learning and artificial intelligence that is rapidly growing in this time. So at the crossroads of these three together, you often see this genius depicted, or the unicorn, or the one who masters it all. And I don't know whether such geniuses truly exist, but definitely it's something that we believe at KeyGene, that the real data science unicorn is in fact teamwork. So interdisciplinary teamwork, where our people enjoy to collaborate and bring together all their complementary expertises to crack the toughest problems and grasp the coolest opportunities in crop innovation. So let me give you an example here. And in this example, we look at root knot nematodes that infect tomato roots and there form these gall-like structures in which they grow and reproduce at the expense of the host plant. Root knot nematodes still cause great damage in the production of not only tomatoes, but in a wide variety of crops. The primary aim of the endeavor that we are undertaking is to help the plant's immune system to solve this problem by itself, so to avoid the need of pesticides to fight the pathogen. We start with collecting and sequencing the genomes of both the host plants and the pathogen, and then we collect and map the variation. We interpret the sequences using our genome annotation engine, and finally we study the gene expression during this infection process with carefully designed gene expression experiments. And here I want to take a little bit of a sidestep for a moment into this genome sequencing and analysis platform, because here our bioinformaticians, our technicians at KeyGene, and our data scientists have put together a really very powerful platform to do genome analysis, where we believe that we can analyze and interpret collections of genomes at any scale. It starts from our particular expertise in high molecular weight DNA sequencing, DNA isolation and long-read sequencing, into a computational pipeline of completely redesigned algorithms for genome assembly, whole genome alignment and comparison, and pangenome and variation reconstruction, and capturing of all this information ultimately in the CropPedia data platform, about which my colleagues Marcel van Verk and Camille de Jong from our partner Ontura will tell you more about in a moment. So back to our problem of the nematodes. After gathering this foundational data, our teams of trait specialists, experimentalists, and data scientists, they get together and they really start crunching and analyzing those data with a suite of data analytics techniques of our lead discovery platform, KeySeq, and they come up with so-called candidate genes. Candidate genes that are involved in this resistance process against nematodes. These candidate genes are subsequently subjected to mutagenesis and knockouts to study their effect on the phenotype precisely. And here again, we make use of automation using deep learning approaches, for example, to accurately and quantitatively assess this phenotypic effect of the mutations. And this then finally leads to a list of validated genes in the crops. So in summary, in this example, I hope that I showed with, starting with knowledge of the trait. We collect and interpret genome data, we design experiments, we perform the data analytics on these data, and in this we, as I mentioned, strongly rely on data science methods in the process. And finally, we were able, in this case, to identify genes involved in the immune response of the tomato plant that helps it in its fight against the pathogen. But to zoom out even further, we have a broad technology suite in place to build genome sites in general, starting with revealing this information. And in these boxes, I list the underlying technologies deliberately in small fonts so that you can read them afterwards and consult our specialists for further information. From revealing the information to understanding the variation, where again, data science plays a crucial role, and with that, exploiting the variation and so enabling downstream applications of trait discovery and genomic breeding. And here, with these latter applications, we support our partners, and here I want to relate back to where we started in the beginning of this presentation, to help our partners to meet the challenges to innovate their crops and ornamentals faster, better, and in a sustainable manner. And with that, that is the story about data science at KeyGene. We hope that this was an exciting talk to you, and when you have questions about how KeyGene's data science can help you or about specific applications, we invite you to please contact us at this email address. So now it's time for our guest speaker who is joining us live from California, Andrew Carroll from Google. Andrew is product lead genomics in the genomics team in Google AI. His team develops methods to understand genomic data and combine it with non-genomic biomedical data. So you can think of applications in like improving clinical genomics and drug discovery. Andrew has a strong focus on genomics community engagement, collaboration and partnerships. And KeyGene collaborates with Google on migrating our heavy computational workloads for genomics to the cloud. In this talk, Andrew will present work on human genomics and other organisms.
Thank you, Roland. Can you see my screen?
Yes.
All right, I'm going to go ahead and begin. Thank you for the introduction. So in this talk, I'll be going over a number of the technologies that our team builds. First, I'll give an overview of a method called deep variant, which is initially trained and validated in human data. And then I'll be talking about two extensions which take deep variant and apply it into sets commonly used for non-human discovery. So first off, in the sequencing data life cycle, a very common analysis is to take the raw output of a sequencing instrument, to map it to a reference genome, and then to identify those positions which distinguish a sample from that reference genome, which is then used downstream, as Roland mentioned, for breeding analyses or in the human space for clinical reporting and drug target discovery. In this process, there are many components which are partially unknown, as well as fully unknown. And I've tried to highlight a couple of these. So when a human is trying to write a tool to analyze this, they have to try to take into account all of these factors. And in addition, they have to take into account factors which humans don't yet entirely understand, which makes the development process quite challenging. So one new approach are these deep learning models. The way deep learning models work is they operate on data in a more raw manner, and they have a series of neural networks that have connections between them, which are trained based on whether or not the neural network training architecture got an example right or wrong. So in this case, it's just a demonstration of this applied to image recognition, which is take a set of cat images, take a set of dog images, present them to a training network with labels, and this classifier will learn for itself those components which are necessary for prediction. And this is one mechanism through which a human who uncompletely understands a problem is able to build a method that can actually account for and analyze those unknown unknowns. So what our team did was to take these infrastructures and to apply them into the genomic space. So for this, we take the raw sequence data and we encode the components of it in something which looks kind of like an image, but isn't really an image. It's a multi-dimensional set of the sequence, base qualities, mapping qualities, strand, and so forth. And using these infrastructures that were developed to analyze things like image data, we can generate classifications that for each position in the genome will say whether this position is homozygous reference, heterozygous, or homozygous variant, and with the confidence associated with it. And you can see on the left, there's this compression of the channels into a kind of false RGB, which is one way that humans can look at the input that deep variant is seeing. So the first proof of concept of this was a submission to the Precision FDA Challenge in 2016. This was one of the very few times when there was a very well-characterized genome that was fully withheld. So participants were given this genome, which was very widely used for development, NE12878 or HE001. And they were asked to produce calls both for HE001 and for this withheld genome. And then at the end of the competition, the withheld genome results were made public, and we were scored against what we had delivered. And this was the first time there was a deep learning method submitted to variant calling, and quite surprisingly, the deep variant, the proof of concept, actually won the highest SNP accuracy. So this was a very good validation that the technique could work in theory. So since that work, we've put a lot of effort into open sourcing deep variant and to continuing to improve it over time. So what you see here is a timeline of our releases as well as the improvements that we have made. And this tool is fully open source. It is widely used. You can go to this GitHub link and download it yourself and run it on your data if you like. So two of the components that I will highlight about deep variant. The first is that deep variant is accurate. So what I'm showing you here are two versions of deep variant compared with state-of-the-art tools, just looking at the false positive and false negatives in a 35-fold human genome. And you'll see that both deep variant has a far lower number of errors that it makes. In addition, you can see that we've continued to improve it over time. And we're now, this slide is, I guess, these charts are a couple of years old. We're now at deep variant version 0.10. So we've continued to improve this with each iteration. And furthermore, you can look at deep variant's estimated quality score, the probability that it believes that it's made an error on a site, and the observed quality score in order to see how often when deep variant says it doesn't understand variant position, is it correct? And you'll see that deep variant is much better calibrated than other methods. And I think that this can go into capturing some of the unknown unknowns in this deep neural network. All right. So now I will shift gears to talk about adapting deep variant for PacBio Hi-Fi data, which is this new highly accurate and long-read technology from Pacific Biosciences. So both Illumina sequencing by synthesis and PacBio are consensus methods over time. The Illumina is a physical consensus that sequences a cluster. The cluster goes out of phase, which limits the ultimate read length. Whereas PacBio, the Hi-Fi sequence is a consensus over time. And this has some advantages, especially in that it can allow for substantially longer read lengths. So one aspect of the PacBio data is that it has a different error profile. The SNPs are highly accurate, but it can have some issues in homopolymer regions of the genome. So what we did was we took labels for the truth set, and we were able to retrain deep variant by showing the PacBio examples with those labels to produce a new model. So we were able to see this comparison on the PacBio data, just applying GATK, which is a standard tool for this analysis, probably the most widely used one. with deep variant, you can see that when you retrain on this data for SNP calling, there is some improvement on SNP accuracy, but there's very notable improvement for indels. So as I mentioned, the error profile for indels is just very different, and GATK is only able to achieve about an 80% overall F1, and we can get that up to 95% just by retraining with the data. And subsequent to this initial analysis, we've gotten these accuracies for indels up to 99% as well. So the last story that I'll tell is about taking deep variant and applying it to non-human genomes. And this is work that was done in collaboration with Malaria Gen, which had a pedigree of sequenced mosquito genomes. So mosquito genomes are quite different from human genomes. In this particular case, they're much smaller. We don't have a gold standard set available. And very importantly, mosquito genomes are much more variant dense than humans. Humans are relatively variant depleted, kind of much more homogeneous in genomic variation. And this makes analyses, both mapping and variant calling, quite different as well. And this occurs in part because of the fast generation time and large number of offspring in mosquito populations. So in order to try to capture some of the additional challenges in mapping and variant calling, one of the things that we did was to generate silver standard data. We had a pedigree of 11 siblings, I believe, and 2 parents. And we were able to take the confidently called positions in the mother and the father and to identify a set of true labels in the child and then train on the child and apply it back to the mother and the father genomes. I should point out that for all the evaluations that I'm showing, what we do is we hold out full chromosomes that have never seen training data to account for overfitting, and we do the evaluation on that. So this model is able to achieve high accuracy. What I'm showing here is a plot of Mendelian violations that are ranked by, on the left, the highest confidence of the variant. So you're able to see that we're able to call much more confidently as measured by this Mendelian violation rate. And in addition to that, to call the most confident variants are much more confident with an error rate that's almost 0. There's a lot that I wish I had more time to tell you about. Hopefully I'm pretty much on time. Our team keeps a blog of all of these analyses. You can check out our tools at GitHub/Google/deepvariant. You can check out the blog at the link that's listed. And I think the many scientists and engineers who have been developing this is actually only a small part of what our team does. Maybe some other time we'll get a chance to talk further about the work. Thank you very much.
Thank you, Andrew. Exciting work showing the power of data science to enable genome insights. Our next speaker is Marcel van Werk. Marcel is team leader crop data science at KeyGene. And Marcel's passion is to use data science to make plant breeders' lives easier. And today Marcel will give a demo about CropPedia, which is KeyGene's data integration and visualization platform. And that is exactly what this platform is aiming at. Marcel.
Thank you, Roland. What I have here and showing you now is the landing page of CropPedia. CropPedia is a data integration platform developed towards breeders to make insights and data-driven decisions. It is hosted fully in the cloud, which makes it easy accessible for anyone, even without any bioinformatics backgrounds. It can rely and host public data, but above all also host your proprietary data. This makes that all your data that you're interested in is integrated in one environment, which I will show you around in this demo. Roland was just talking about actually cyst nematode resistance. So if we in this CropPedia, here in the main search bar, look for cyst. Further, actually, we also have quite some tooling available. So we have the search bar and we have a genome browser, comparative map viewer to show your genetic or molecular maps, a variomics viewer, which I'll show you later in a bit more detail. We can browse the data in detail. We can look at blast alignments. And lastly, we have an omics analysis. So now go back to the nematode resistance. So we take the keyword cyst, and actually we look what CropPedia finds on all of this. So it finds, as you can see, a number of genes. It finds further quite some locations within the genome. Further, it also looks into public data, public domain data, papers. There's no pathways directly that are linked, but also pathway information is loaded within the CropPedia, so we also have insights into that. But for example, if I look in this rice line, I see here already one gene that says cyst nematode resistance. So if actually I look a bit further into that one, I open the so-called gene passport within CropPedia, and this passport contains all information you can basically almost need that has information about this gene. Accession information, at the same time also the coding sequence, protein sequence, information about the localization, alignments with other proteins, protein clusterings, and it also shows you, for example, for this particular gene and all the variant data sets that have been loaded, at each of the positions, which variants are available in which lines. So you have directly a full overview of all variants available from your resequencing information in one environment. This data can readily directly be exported into Excel, which makes it directly available for any downstream application. Further, if there's expression data loaded, you also see directly expression profiles. For example, here is some resistance to different bacteria. And you see that under certain treatments, also this resistance gene, which probably has something to do with cyst, also shows very high expression upon treatment with this bacteria. And then another link again that this gene in general has something to do with resistance in the plant. If I take it now a bit further and actually I go look into the variation data, I can go to the variomics viewer. The variomics viewer is capable of actually visualizing variation data at a genome-wide scale. So here on the left is a very large amount of resequencing lines that I can load all, and actually it will on the fly retrieve all variation data at a genome-wide scale and generate heat maps for each of those resequencing lines. So here you directly have a huge overview of all the variation you have available in your different lines and your different wild material, for example. You can make selections here. For example, if you want to select a set of lines that are resistant and you want to select a set of lines that are susceptible towards cyst nematodes, then actually it will show you the contrast between those and highlight only the SNPs that are different between those lines, directly integrating and giving you direct leads towards novel goals towards breeding. Another part which you can research within the CropPedia is using the omics analysis toolbox. The omics analysis toolbox is capable of visualizing expression data as well as metabolite data. And in this example, some expression data has been loaded. So first of all, I will just search for some genes with at least an expression value relative of 50. It will live retrieve all the genes that under all kind of different conditions fulfill this requirement. Now I'm going to select just the top 100 genes. So at least I know there's quite some expression. And I'm going to state analyze. You can also export this directly also to Excel for further downstream applications. But in this case, I'm going to analyze the data. And I want to see the correlation of all these genes relative towards each other. And I'm going to select this bacterial resistance as a comparison. And on the fly, it will generate you a correlation matrix where actually you have information about how all these genes relate. You can live zoom in, zoom out, export it, see the raw values, etc., providing you further insights, how genes relate, and what you can actually do with them. Another important part actually of CropPedia is that you can load your proprietary data. So we have an administrator panel. And in this administrator panel, you can control anything within CropPedia. It can control which users can make use of the platform. You can make groups of certain users, for example, are only allowed to see tomato data, while other ones, for instance, rice data. So there's all kind of levels of security in there. There's an entire documentation about API calls, and that makes that anything that you see within CropPedia can also be automated towards your bioinformatic pipelines in-house. Another handy feature is that here you can see for any user the data management, and you can get a full overview of all data sets that have been loaded within CropPedia. And the handy part is also you can select some of these data sets. If you scroll down, you get directly all information about these data sets, and you can retrieve all the raw data sequences and all the raw information directly downloadable for any downstream application again. So I hope with this, I hope that I showed you that CropPedia is really a data management platform for your proprietary and your public data, integrated all together in one environment with very advanced controls, making your life as a breeder easier. And with that, I give the floor back to Roeland.
Thank you, Marcel. Of course, that all sounds pretty amazing and good stuff, but the proof of the pudding is in the eating, and that is why we have invited our second guest speaker, Camille de Jong from Ontura. Camille is Manager Molecular Applications and one of our partners in the ornamental breeding industry. Camille will introduce Ontura and will explain one of the applications for which Ontura is using CropPedia to improve the breeding of their ornamental plants.
Thank you, Roland, for your invitation and the opportunity to tell something about the work that we do at Anthura. Well, first I will start with a small movie about Anthura as well. Everybody knows what Google is, but not everybody in the audience, I think, knows what Anthura is.
Anthura is the world leader in breeding Anthurium and orchids, a company of innovation-minded entrepreneurs, unlimited in varieties, offering unlimited possibilities. This is what we stand for. Every day, unique, colorful plants are created and produced all over the world. We believe diversity drives innovation. Our R&D department has sequenced and mapped the DNA of Phalaenopsis and Anthurium. In our ultra-modern research center, our experts elaborate on this unique milestone in horticulture. Anthurium and Phalaenopsis tissue culture is made in our own lab facilities in the Netherlands, Macedonia and China. Our state-of-the-art production locations contribute to the most sustainable production processes and products in both the Netherlands and Germany. We contribute to biodiversity in our environment and respect natural resources. Working together, we are Anthura.
Presentation. Well, I will first say something about the strategic collaboration we have at KeyGene and Anthura. At KeyGene we have a compact team that is focused on trait development for our crops, and we need to make clear choices about what we do ourselves and what we can't do ourselves. So here KeyGene comes in. We can make use of the experts of KeyGene. There's about 150 experts working at KeyGene, so they help us to build up and support us with the molecular basis of our crops. And make it available and accessible for scientists in diverse disciplines within Anthura. The milestones of the collaboration include both the genome sequences of the commercial varieties of Anthurium and Phalaenopsis in this collaboration. Why we use CropPedia? The CropPedia makes genomic data available and accessible to our scientists from various disciplines without their having to know very much about bioinformatics. The bioinformatics that is performed upfront by KeyGene and implemented in the databases of CropPedia. But as Marcel just showed, you can also put in data yourself or download data from this database if you need to. Well, also what is useful for us is that KeyGene can also put in publicly available data. But the data that is available for our crops, it requires, well, to put it mildly, some polishing to be usable in this kind of platforms. And KeyGene does that for us. Well, also, as Marcel already told you, you can work in a proprietary environment without you having to upload your data to a public server or so. So that's very comfortable, also for IP issues. The maintenance and support is also done by KeyGene, and that's also very convenient for us. So we don't have to. We can focus on our traits instead that we have to focus on keeping a genome browser into the air. What's also is very useful is that it's cloud based. Nowadays, you can choose if you want to have it cloud-based or not. For ICT, it's very convenient because it's secure, and you don't have to. You can adjust it also to your needs. If you have bigger data, it's very easy to increase the size of your platform without having to add additional iron, as they call it, and to size it up. So that's for us is also very convenient. Well, in this talk I will focus on Phalaenopsis and not on Thurium. To say something about Phalaenopsis. Phalaenopsis, it's an autotetraploid. It is a genome size of about 1 gigabase pairs. It's a polymorphic outcrosser, which makes well, genetics quite complicated. There are some publicly accessible genomes available, but they are of various qualities. Some are quite good, but others they are highly fragmented. There are about 12 species introgressed in the commercial germplasm, which also makes it highly polymorphic. KeyGene sequenced a proprietary genome sequence for us of the tetraploid commercial variety. Here I will have a showcase of a marker. There was a QTL analysis done, and we found an interesting marker of an interesting trait. But it was not convertible to an operational marker, and those are PCR based. For those of you who are familiar to work with diploids, well, this is something of a change, because instead of 3 small clusters, you see about 5 clusters in this plot, and that is also the dosages that are encountered for the various markers that are genotyped in this array. Well, as you see, for this cluster, it went very well in the array, but not very good in the PCR-based operational assay. So we had to see what was going on there. But first, why would you invest so much time to convert a marker? Well, if a marker has a high lot score, it means that the marker is very valuable, because the high lot score, it's important because of the linkage disequilibrium decay in your germplasm. So you very easily use your coupling of your marker in a wider germplasm. So you want to have it as close as possible towards your trait of interest. Also, especially in outcrossing polyploids, the chances are very low that you will identify another marker that is correctly phased. If you look at it in a biparental population of an outcrossing autotetraploid, you have to take 4 different alleles of each parent into account. So these are 8 alleles in a population as compared to 2 alleles in, for example, a conventional backcross in a diploid. Well, the first step that we take in that is to blast the RefSec of the marker in the array against the genomes that are present in CropPedia. Sorry. Well, if you look at the blast result, what you see is that this RefSec it fragments in 2 parts. It comes into 2 loci, so you see that there must be something amiss. So what you also see is that there is a clear SNP there, which is recorded as the mismatch in the blast result. If we click on the blast result, we immediately go to the genome browser in CropPedia, and there you can activate the relevant tracks that you need in your analysis of that. So what we see here is on top the track that is the blast result that you see. Under that we see the gene prediction that was also done by KeyGene, and below you see the SNPs that were present on the array that you also are clearly present see in that track, and also below that there's another track that shows the reference sequences from the GMAP array, and those are highlighted in pink. Below you see also some transcriptome data that are also present, and that collaborate with your gene prediction that you see above there. So if we zoom in there, what you see is here the 2 pieces of your reference sequence that end up on different places in your genome. And what we see here is that the reference sequence of the array is stretching over an intron-exon junction here. So that is possibly what caused the PCR-based assay to fail here. Well, the SNPs on the array, they were mined on transcriptome data, and we are looking here at the intron-exon junction. And the intron interfered with the PCR-based assay. In contrast to the PCR-based assays, in an array you can call a SNP from one side, and that's not possible with a PCR-based assay. So the SNP can be perfectly all right on an array, but it does not convert to a PCR-based assay. During that design of the array, we checked for. We did it in collaboration with the WER, and we checked for the intron-exon boundaries. But at that time, there was no good genome available, and that genome was highly fragmented. So that caused the filtering, I think, to fail in some places. Well, what you can do to remedy that is expand your SNP reference sequence by downloading the ruler sequence from the genomes that you look into in Encropedia. And you can very easily do that by clicking by the mouse and select the area you want to, and then you can download the reference sequence on that area. Well, what you do then, after having expanded your reference sequence, you will make an alignment there, and you can see where in the reference sequence you have to take into account other SNPs, but you also see here clearly the intron-exon junction and the target SNP in this alignment. Well, then we developed a new PCR-based assay, and you see there on the slide that the PCR-based marker did work in one population. That is the third square that you graph that you see there with the clusters, and you see on the whole the slide on the left there are multiple populations, so you see multiple clusters, and the marker now clearly worked. So the problem here is solved. Well, thank you for your attention, and I'd be happy to answer any questions.
Thank you, Camille. Indeed, we are proud that CropPedia is supporting Ontura in such a concrete and important way. And that brings us to questions from the audience. If you haven't typed in your questions yet, I would kindly ask you to do so in the chat box. But before we start answering your questions, I would like to ask you to fill in a brief questionnaire about this webinar. It is only 4 questions, and your answers will help us in improving our KeyGene webinars. And you will find a link to the questionnaire in the chat. So let's have a look at what questions we already have in the chat. I see a question from, let me have a look, from Charlotte Prodom. I think that is to Andrew about deep variant. Andrew, do you plan to adapt deep variant to polyploid genomes?
Yes, it's a good question. You can hear me, correct?
Yes.
Okay. So I think that, so yes, this has come up. I think that we are evaluating the right strategy to take, and this will depend on how completely the reference genome can be assembled. So if we think that the polyploid species are going to be able to be basically phased and then assembled in a manner that the reference will be complete and have a full diploid on it, then we would take one training strategy. Whereas if we think that the reference will be compressed relative to the true content, we'll take another strategy. So we haven't done this yet. I think we'll probably follow the lead from the long-read technology companies that we collaborate with, Pacific Biosciences and Oxford Nanopore, about the best strategy to take. So I know this is one of the hardest problems. So we haven't done it yet, and I would say look forward in the future.
Thank you, Andrew. And that maybe nicely bridges to a question that we have from Nick, and which is related to this. Does deep variant work on ONT data?
Actually, very recently, we collaborated with UC Santa Cruz. They actually have developed this method for generating candidates in Oxford Nanopore data that we then can feed into deep variant. And so we trained a model, and actually we're able to show superior SNP accuracy relative to Illumina on a human data set. And the group that we're working with at Santa Cruz is Benedict Patton's group, and the principal investigator was Kishwar Shafin. If you Google Pepper, P-E-P-P-R, deep variant, you should be able to find the GitHub link. You can run this yourself right now on Nanopore data in sort of an early, limited release manner, and we're working to make that a full release as well.
Thanks again. But there is another question, Andrew, from Irina. She thanks you for your nice presentation. And she asked whether you are interested to try deep variant on very large and complex genomes, such as conifer trees, which have an enormous size, and which have highly repetitive genomes?
Yeah, I think this is a very good question. So to start this answer, I don't know the quality of the results that you're going to get applying pretty much any variant calling method, meaning I don't know how things like freebase will behave. I don't know how things like JTK will behave. I'm not sure how deep variant will behave. Mechanistically, it is certainly possible to do so. And I think that you'll likely get mostly valid results. I think it's something that we would have to do and take a look at, and then potentially to retrain for some of these sets of different crop genomes. So highly repetitive is one class, extremely large genomes, relatively incomplete genomes, polyploid genomes. I think there are these sets of types of plant genomes, and it might be good to have like one type of model for each of those categories.
Thank you, Andrew. I think then we switch gears a little bit to Ontura. Again, a question from Charlotte Prodom to Camille. Is the 4X Phalaenopsis genome sequenced by KeyGene phased?
No, it's a good question. It's not phased.
It's not phased yet. And another question, I suppose that's also to you. How much closely linked SNP markers will be used in your breeding program?
Well, that's yeah, it's a difficult question to answer that because
maybe we
it's
give you a
also
little bit of time.
uh,
We give you a little bit of time to think about it, and we'll get back. So, how many close linked marker will be used in the breeding program? And we get back to you, Andrew, with a question from Ertu Grul. Can we use a deep variant to discover SNPs in genotyping by sequencing data?
Right. So this depends on some extent to the coverage that you use. So, if we're talking about 5-fold coverage, it's in theory possible to do so. And in fact, we have done titration experiments that show that deep variant holds up in accuracy much better at 5-fold coverage. If the coverage is lower than that, if we're talking about like 1-fold coverage and you're trying to genotype markers, we're currently working on mechanisms to do force calling and to have models that are trained for this, but we don't have these readily available. So hopefully over the course of the next year, we'll be able to get to that point. But right now, with what you have out of the box, coverages below, say 5-fold, are unlikely to work well.
Okay. Thank you, Andrew, again. I don't see any further questions. I don't know if you cracked the difficult question that you got, Camille, in the meantime.
Yeah, well, I was thinking how to interpret the question because it depends. You have in your breeding program you have different subsections. So if you want to use certain subsections, I think there you have to apply different kind of markers in that, and also how many markers do you need to if you want to select for seedlings, for example. Well, if for example you can throw away with one marker, you can throw away 20% of your sequence or your seedlings. Well, at 5 markers, you have nearly anything left. So that's more or less. I hope this answers your question, but that's a bit the thing that we also are considering. How much do you want to throw away at the end?
Thank you, Camille. I hope that answered your question, Anand. If I now have a look at the chat box, I don't see further questions coming in. So, also looking at the time then that we are, Andrew's working day is now starting, and here in Europe we are getting hungry, so in for a meal or a drink, and maybe in Asia it's almost bedtime. So with that, I would like to end this seminar. Again, thanking our guest speakers, Andrew and Camille. I think your presentations indeed showed very nice progress and a little bit of the perspective ahead in using data science. I also want to thank the audience. Of course, after all, we organized this webinar for you. I hope that you enjoyed the seminar and that it was worth your time. Again, please do fill in the questionnaire before leaving the webinar. Your view, again, as I mentioned, and your suggestions will be very helpful to us to improve our webinars. And with that, I wish you a good day or a good night to all of you. Take care. I hope to meet again soon, digitally or in person. Thank you.